In this chapter, we're going to take a look at bacterial growth and see what it means when we talk about that term growth, and then look at the phases that a culture or colony of bacteria might go through. So microbial growth is less about an individual changing size than it is about the total number of bacteria that are found in a colony. Because bacterial cells, regardless of their age, are generally about the same size. So, sorry about that, I just dropped something there. Okay, so the bacteria do grow, so individual bacterial cells do grow, but as soon as they roughly double their size, then they immediately divide, go through a process called binary fission, producing two daughter cells, and then the cell begins to grow again and then divides again. And so we don't really get concerned about the actual size of any individual cell, because as soon as it gets to a little bit bigger, it's gonna divide. And so when we talk about bacterial growth, then what we're talking about is the increase in the number of cells that are found in a broth culture or on an agar surface. That's what bacterial growth is the increase in the size of the entire colony or culture. So the process that bacteria use for growing is not mitosis like we see in eukaryotic cells, but instead is a process called binary fission, or sometimes we see budding in cells as well. So in binary fission, a cell increases in size, the chromosome duplicates, and the cell continues to increase in size. And then we see the development of a septum, which is a chunk of cell wall and cell membrane that begins to grow and separates the two daughter cells and their respective chromosomes from each other. So if we have, let's say we're looking at a rod-shaped bacteria, and here is the nucleoid region with its circular chromosome. The chromosome is found in an area, the nucleoid. We've talked about that in the previous chapter. And the chromosome is generally attached to a little area of the cell membrane. And when the chromosome duplicates, the new chromosome is attached in a slightly different area. And so as the cell grows, the regions where they're attached move further and further apart until a septum can then grow in between them. And so a septum is a chunk of cell wall with the membranes on either side of those two chunks of cell walls, and then once that septum is fully grown, then we see that there are now two different cells. So hopefully that makes sense. But the big thing to keep in mind is that the chromosome once it divides, is the new chromosome is attached in a slightly different location, so that as the cell grows, the chromosomes grow apart from each other. So they're found now in two distinct regions, and then all it takes is a new chunks of cell wall and cell membrane to divide the two bacteria from each other. And here we see binary fission in a cockle, in a coccus, in the picture that I found, and you can see it dividing. So we have a large cell that then divides into two smaller cells, and you can see in this last image here that they are fully separated from each other, and the cells will then grow to reach the size of the parent cell. before it divided. Okay, so there is not always complete separation of the cells, and when the separation is incomplete, then we see the development of all the different arrangements of cells that we can see in bacteria. So here in Streptococcus, we can see that we get chains of the cocci. In Streptobacilli, we can see that, and here our example is Bacillus megaterium. We can see that the bacilli wind up being linked together in chains. Sometimes they form these tetrads where 4 cells are arranged together, sort of in a little square shape, like we see in Deinococcus. Or they might form Sarcini, where we get these cube-like clusters of 8 cells, like we see there in Sarcina. And then last of all, the grape-like clusters that we see in Staphylococcus. So that occurs when there's incomplete separation following binary fission. And the arrangements of the cells can be really affected by the nutrient levels and the kind of medium that the cell is growing on. So whether they do this kind of incomplete separation can be a function of the nutrition that's being provided to the colony. Here we see yeast cells. Yeast cells and some other bacteria go through a process called budding, where new little small cells develop at the surface of the parent cell, and as they become larger and larger, then they will pop off and disconnect from the parent cell. And so those little cells are referred to as buds, and that process is called budding. And budding is super common in the yeasts. Okay, so now what happens to an entire culture of bacteria as they are growing? As you think about this process, imagine that we take a small loopful of bacteria from an old culture and introduce that loopful of bacteria to a new culture, a set of new media where they will be growing in a very nutrient-rich medium. And there are 4 phases to what we talk about as the standard bacterial growth curve. So 4 phases of growth that occur. The first phase is called the lag phase, and then it goes through a log phase. Then for a time there will be a stationary phase, and the stationary phase will be followed by a decline or death phase. And the thing to notice here is that once the population has adapted to the media that it's living in, so if you take bacteria from an old culture, those bacteria have become adapted to a nutrient-poor medium. And so once the bacteria have adapted to being in a nutrient-rich medium, then population growth can be very, very rapid. And that contributes then to what we talk about as the log phase. So let's begin then with lag phase. During the lag phase, this is a time where there's not a lot of growth in the colony or the culture size. But that doesn't mean that the individuals aren't involved in metabolic activity. So metabolic activity is there's a lot of it going on with the individuals. And during lag phase, those individuals are increasing in size and producing lots and lots of activity. And how long this lag phase lasts really depends on the conditions that are in the culture media. If they're very different from where the bacteria have been taken as they're introduced into the new culture, then the bacteria are going to take longer to adjust, perhaps. As well, different species of bacteria take different amounts of time to become adapted to new media. And if they've been starving where they were, then it may take a while for the bacteria to become adapted to that new medium that they've been placed into. So if the bacteria are coming from a medium that is really, really rich in nutrients, then that lag phase may last just a few hours. So it really depends a lot on the circumstances of the old medium and the new medium that the bacteria are being introduced to, the condition of those bacteria as they're being introduced, and then what species it is, because every species is going to have a slightly different response to being introduced to new medium. So lag phase, not a lot of change in the number of cells because the cells are busy adapting to their new medium. Log phase, then, is a time of very rapid growth in the number of cells that are found in the medium. And I want you to note that the y-axis here, it is on a logarithmic scale, and so each, if we were to add some units here, what you need to know is that this is not a linear scale here, but this may be, if that's 10 bacteria, then this next little hash mark is 100 bacteria, and the next one is 1,000 bacteria, 10,000 bacteria, 100,000 bacteria, and so on. So this is on a logarithmic scale rather than on a linear 10, 20, 30, 40 scale. So what this is saying is that every unit that we go up is actually a multiplier of 10 rather than being an adding 10 to it. So logarithmic growth is super rapid growth. And so if we think about a bacterium that has a doubling time, so a bacterium that is where the cells are dividing approximately once every 20 minutes, and we started at our 0 hour with 100 individuals, by the time we get to 1 hour, that's 3 population growths later, notice that it's not 3 times the individuals. We haven't gone from 100 to 300. Instead, the population has doubled with each of the intervening times. So we've gone from 100 to 200 individuals, from 200 then to 400 individuals. So by the end of an hour, there'd be approximately 400 individuals in our medium. By the end of 3 hours, there'd be approximately 25,000 individuals in the medium. And by the end of 8 hours, and this is just mind-boggling to me, but there would be more than 8 trillion individuals in this growth medium. So logarithmic growth is very, very rapid growth. For most bacteria, a generation time is going to be something on the order of 20 minutes all the way up to 20 hours. So every bacterium has a different generation time at maximal growth. And so it's going to be somewhere between 20 minutes and 20 hours. But the typical bacterium, so the average bacterium, is going to have a generation time of less than an hour. So 20-hour generation times, that's a really long generation time that we see. Okay, so hopefully logarithmic growth makes sense here. And their growth is what is referred to as non-synchronous. So it's not that there's a clock, and it goes ding, and at the end of 20 minutes or at the end of an hour, all the bacteria, boom, divide at the same time. If that were the case, then we would see this red line we would see a very stepwise growth in bacteria numbers in a colony. And that's not what we see. What we really see is more of this green line where, every bacterium is on its own little schedule, and so it's more of a smooth curve showing growth. We see non-synchronous growth because everybody's rate of binary fission is slightly different, and when one cell divided, another cell was still in process, and so that means that the numbers, if we were to sample every minute and we could somehow get a count in the colony in a cell that had a 20-minute generation time, if we could sample every minute, we would then produce this green line curve where we see a much smoother rate of growth rather than a stepwise curve like you see in the red line. Okay, so hopefully non-synchronous growth makes sense here. Okay, so why is it that what brings an end then to the log phase of bacterial growth? Because it can't be sustained forever unless you have a system where you're constantly supplying fresh medium to your colony and constantly removing old depleted medium. So if you did that, and there are machines that will do this, then you could maintain the log phase indefinitely by continually supplying fresh medium to the bacterial colony. But instead, if what we see then is instead the lazy microbiologist or the realistic microbiologist, over time, nutrients available for growth are going to be reduced, and that's going to limit the rate at which colony growth continues. So as nutrients get used up, then colony growth is going to slow. And along the way, as nutrients are being used up, then waste materials are going to accumulate in this broth culture as well. And waste material accumulation can also slow the growth of the culture. There'll become less space available for the bacteria in the culture, especially those that grow on a surface. So that makes sense, hopefully, if the bacteria need a surface to grab onto, then over time that surface is going to be filled up with more and more and more bacteria, and there'll be less space for growth to happen. Oxygen can become depleted in the culture as well, and so if we have an aerobic bacterium, one that requires oxygen for growth, then we're going to need to bubble in oxygen so that the colony doesn't decline too quickly, because there isn't enough of the oxygen for them to continue growing. And over time, as waste materials build up, many of those waste materials that bacteria produce and all organisms produce are acidic, and so the pH in the medium can also change and become not ideal for the bacteria. So lots of reasons why the log phase can't be sustained for a long period of time, and it's all about they just run out of some limiting factor, or they force the conditions in the colony to be outside of what is ideal for bacterial growth. Okay, so as conditions degrade in the culture, then the rate of growth changes, and we see a leveling off happening in the number of cells that are found in that culture. And this occurs because the rate at which the cells are dividing is going to slow. And eventually the colony reaches the point where the new cells that are added are going to be offset by the number of cells that are dying at any time. And so at this point, the size of the population becomes stable, and we enter into what is known as the stationary phase. stationary because the size of the colony isn't moving anywhere at all. And then as conditions continue to degrade in the culture, they can become so poor that the cells lose their ability to divide. And just as rapidly as the cells go through the log phase, they can then go into a decline phase as the colony begins to die. So we see that the size of the colony decreases now at a logarithmic rate, and we see relatively speaking, a rapid decline, almost as rapid as the growth phase was. And there are some interesting things that happen in the colony over time. One is that the cells undergo a process or a change in their shape called involution. So as the conditions in the colony degrade, then we start to see changes in the shape of the cells and in their staining qualities too. And spore-forming bacteria, this is the point when many of the cells begin to, or not only, they may have been making endospores for a while, but as they die then, as the cell dies and lyses, then the spore is released, and now that individual, its DNA is no longer going to be actively causing active metabolic work to go on, and the cell enters a resting stage. And eventually, over time, all the cells will either die, if it is a colony that can't form spores, or a bacterium that can't form spores, or the cells all sporulate, and as the vegetative cell lyses, then we're left with the resting stage. So, depending on the species of bacteria we're working with, the population can go through all these stages: lag, log, stationary, and then into decline. They can do that within just a few days. Or some colonies will, this decline can be very, very slow, and there may still be living cells in that culture after months or even years, because the cells can adapt to their degraded environment and slow metabolic processes so much that the cells can, there can be living cells hoping against hope for a change in the conditions with where the colony is. And we see these same phases occurring not just in broth media, but also on agar plates. And so, if here's a bacterium that lands on the agar plate, and as it grows and divides, then we see, the colony becoming larger and larger, and the original region may be in decline, while out here at the edges, the colony may be just entering the log phase as it encounters fresh medium and grows larger and larger and larger on the plate. So it's not just cultures that are in broth, in nutrient broth, that undergo these stages of lag, log, stationary, and decline. It's also cultures that are being grown on solid media too.